To everyone. So yesterday we are discussing flower. In flower we are discussing parts of the flower we completed. So now we will discussing one by one sepals, petals and androecium and gynoecium. So we will take androecium. First part it is stamen. Okay. So stamen. So in stamen we will discussing transverse section of stamen completed. So next we will discussing microsporangium. Next we will discussing microsporangium. So microsporangium, first we will draw the diagram and we will explain. Okay. So first we will draw the diagram and we will explain. So this is the mature anther. This is the mature anther. Okay. If you take a transverse section of mature anther, so we will see in so this outer structure we will see in. So outer structure generally it is a circular. It is how it is there, circular outer. Okay. So outer when we will see it is a look like a circular in outer line. Okay, circular in outer line. So generally it is made up of 4 layers are there. How many layers? 4 layers are there. So first we will discussing so first epidermis. This layer it is called as a epidermis. This layer it is called as a epidermis. This layer it is called as a epidermis. Okay. So outermost layer it is epidermis. This is the outermost layer. This outermost layer generally it is epidermis. Outermost layer epidermis. This is the epidermis. So first we will draw this side. Okay. So that same, this side also same, that one only. Okay. So this is the layer, continuously it is there. Okay. This layer, outermost layer it is called as a epidermis. Like that we should draw. Okay. This is the outermost layer. We will draw like this. This is the outermost layer. That is nothing but epidermis. This is the outermost layer. So that is nothing but epidermis. So next it is followed by one more layer it is there. That is nothing but so this is the epidermis. Outermost layer it is epidermis. So next it is followed by one more layer it is there. That layer it is called as a endothecium. This layer, this is the endothecium. This layer is called as a endothecium. This layer it is endothecium. This layer it is endothecium. This is the endothecium layer. It is followed by next layer, that is endothecium. Is followed by next layer, that is endothecium. Okay. So in this all the sides same like this only it is there. First we will draw and we will explain. So one side we will draw and so same in all the sides same layers only it is there. Okay. First we will draw. So one side we will draw and we will explain. Okay. So next layer it is middle layer. Generally it is the middle layer. Middle layer generally 2 to 3 layers are there. How many layers are there? 2 to 3 layers. So these are the layers. This layer it is generally middle layer. This layer middle layer, but it is made up of parenchyma cells. It is made up of parenchyma cells. And also here generally in this region connective tissue it is there. In this region connective tissue. So both lobes will be separated by connective tissue. Okay, so that one we will discussing. So how many layers are there here? This is generally so 2 to 3 layers are there. 2 to 3 layers. How many layers are there? 2 to 3 layers. Here total 2 to 3 layers are there. Total how many layers are there? 2 to 3 layers of middle layer it is there. So next it is followed by one more layer, that is tapetum cells. Another layer it is called as tapetum. So this is tapetum. Generally these cells are pyramid shaped cells. Generally these cells are pyramid shaped cells. These are the pyramid shaped cells, like it is there. This is the pyramid shaped cells. So inner side it is called as a sporangium, microsporangium. Here what it is there? Tissue is there. The tissue is nothing but microsporangium. This entire thing it is called as a microsporangium. In center region so some tissue it is there, that is called as a sporogenous tissue. The tissue is called as a sporogenous tissue. Okay, see here also same like that only. So in this also last layer, this layer it is called as a tapetum. This layer it is called as a tapetum. So inner side it is sporangium. Young anther contain sporangium it is there. The sporangium center position tissue is there. The tissue is called as a, so generally this tissue is nothing but sporogenous tissue. This tissue is nothing but sporogenous tissue. This tissue is nothing but sporogenous tissue. So like it is there. So when we will see, so this is all layers are there. Okay, so this is the layers. Okay, like it is there. Okay, sporogenous tissue. So now I will explain one by one. Okay, this is the diagram. Okay, so generally we will see in Indian it is look like so outer line. Okay, so the transverse section, when we will take transverse section, so the anther outer line it is how it is there. It is look like circular. It is look like so circular. Okay, see here it is so outer line, outer line. So how it is there? Circular. Outer line it is circular in outer line. Circular in outer line, how it is there? Circular. Okay, so how many layers are there amma here? So first we will so labeling. So this is called as a epidermis. This is called as a epidermis. So next it is followed by another layer it is there. Okay, so this is followed by another layer, that is called as a endothecium. Endothecium. Okay, endothecium. So next layer it is there. This layer it is called as a middle layer. Middle layer. That layer it is called as a middle layer. So next layer it is there. So that is nothing but so this is the another layer. Okay, so this is nothing but another layer. This is called as another layer, that is called as a tapetum. That layer is called as a tapetum. So next one it is so this is called as a sporangium. Okay, what sporangium? Microsporangium. Okay, so this entire region is called as a microsporangium. Young anther contain what it is there? Microsporangium. Young anther contain only microsporangium. Okay, young anther contain what it is there? Microsporangium. Okay, this is microsporangia. Microsporangia. In microsporangia, what type of tissues are there? Only homogeneous tissue. Okay, so what tissue it is there? Homogeneous tissue. Okay, what it is? Homogeneous tissue is nothing but sporogenous. That is called as a sporogenous tissue. Sporogenous tissue it is there. It contains homogeneous cells. So homogeneous tissue. Okay, homogeneous tissue is nothing but what? Homogeneous tissue. Homogeneous tissue is nothing but so sporogenous tissue. Okay, sporogenous tissue, where it is present? In center position. In a center position. So this is the labeling of microsporangium. So how many layers are there? So one, it is outermost layer. This is the first layer. Next it is second layer. So next one it is third layer. So this is the fourth layer. Okay, total 4 layers are there. Total how many layers are there? 4 layers. So first we will discussing what we will discussing? Epidermis we will discussing. Okay, first what we will discussing? Epidermis we will discussing. Okay, so next one it is epidermis. Okay, so first one it is epidermis. Okay, first one it is epidermis we will discussing. So in epidermis, so what it is the outermost layer. It is outermost layer. It is a outermost layer. Okay, it is a single layer. It is so it is made up of what it is? Thin walled cells. Thin walled cells. It is thin walled cells. It is made up of what cells? Thin walled cells. Okay. So next one it is generally the cells are flatted shape. Okay, flatted shaped cells. The cells are so flatted shape. Okay, how it is there? Flat. Okay, how it is there? Flatted shape. And it is also protective in nature. It is also protective. Okay, it is also protective in nature. It is protecting. It is protecting outermost layer, thin walled cells, flat shape, and protective in nature. Protective in nature. So next we will discussing so second layer. This is the second layer. So second layer generally what it is? Endothecium. Next layer it is nothing but endothecium. Endothecium. So endothecium, what type of cells are there? Okay, it is initially it is a thin walled cells. Okay, so generally it will become a mature thick walled cells, but so in this region thin walled cells are there. Then it is also helps to the opening, then it releases into the pollen grains. Okay, so that one we will discussing. Here what it is? Endothecium. In endothecium it is which one it is followed by? Epidermis followed by epidermis followed by so endothecium. Endothecium. It is also so second layer. It is second layer. It is second layer. So here cells are arranged radially arranged. Okay, the cells how it is arranged? Radially arranged cells. Radial arranged cells. Radially arranged cells. That cells are radially arranged cells. So generally, so the cell, so it is made up of callus. Okay, the cells, what it is made up of? Callus. Cells are made up of, cells are made up of, okay, it consists of what it is there? Callus. Callus is mainly where it is there, cells are made up of callus. Cells are made up of callus. What is the use of callus? Generally callus, it is also helps to the dehiscence of anther. Okay, what it is? Dehiscence. Okay, it is also helps to the dehiscence of anther. Okay, it is also helps to the dehiscence of anther. It is also dehiscence of anther. What it is dehiscence of anther? It is also helps to the opening. Okay, it is also helps to the open. Okay, dehiscence means what? Opening. It is also helps to the opening. Okay, it is also helps to the opening. Okay, so this is also endothecium. This is also endothecium. So next one it is so next layer it is middle layer. Next layer it is middle layer. This is middle layer. Okay, so generally it is a middle layer. Middle layer it is a third layer. Middle layer it is third layer. So middle layer it is third layer. So here mainly middle layer it is thin walled cells. Okay, middle layer it is thin walled cells. Middle layer it is thin walled cells. Okay, mainly it is parenchyma cells. Parenchyma cells. These are parenchyma cells. Okay, so generally it is also storage food materials. It is also storage food. Okay, the middle layer it is also helps to storage of food material. Okay, so this is also we will observing in so middle layer. But these three layers here mainly these three layers, how it is helping? Mainly these three layers, it is also helps to the protecting and also it is also helps to the dehiscence. Okay, dehiscence. Okay, dehiscence, it is also helps to the protecting and dehiscence, these three layers. Okay, according to NCERT, so it is also helps to the so protecting and dehiscence. Okay, these three layers. Okay, these three layers. So next what layer it is there? Tapetum. So next what layer it is we are discussing? This is nothing but tapetum. So tapetum it is a innermost layer. Tapetum is a innermost layer. Tapetum it is a innermost layer. Here I will write tapetum is a innermost layer. Tapetum is a innermost layer. Yes, last layer. We consider as a last layer. Okay, here generally it is made up of what type of cells are there? It is generally it contain what type of cells are there? Here mainly we will see in pyramid shaped cells. Pyramid shaped cells. What cells are there? Pyramid shaped cells. Okay, pyramid shaped cells. So these cells with for example if you take once this is the pyramid shaped cell. So it contain dense cytoplasma. Okay, it contain dense cytoplasma. Okay. So next one it is next layer it is called as a tapetum. Tapetum is a innermost layer. Already we are discussing this is tapetum. It is a last layer. It is pyramid shaped cells. Okay, so it contain dense cytoplasma. Dense cytoplasma. What it is there? Dense cytoplasma it is there. It contain what it is? Pyramid shaped cell. It contain dense cytoplasma it is there. So it contain also, so more than 2 nucleus it is present in. Okay, it contain how many nucleus are there? More than 2 nucleus. It contain more than 2 nucleus. So why it is more than 2 nucleus means so this is the which one it is? Nucleus. The nucleus it is dividing by the so mitosis. What it is takes place? Mitosis. Mitosis is takes place. Then what happen in cell? This is the pyramid shaped cell. What happen? Endomitosis. So what inside the mitosis takes place? That's why it is called as a endomitosis. This is called as a endomitosis. Endomitosis. In endomitosis takes place what happen? So this one nuclei it is converted into the 2. Again it is divided into 2. So again it is divided into 2. So that's why so more number of nucleus is present. Okay, more number of nuclei are present. So it is also tapetum functions. Okay, it is also helps to the formation of calyx. Okay, it is also formation of calyx. Calyx. What enzyme? Calyx. So calyx, how it is helping? Here what it is there? Callus. Okay, so cells it is made up of second layer endothelium with callus it is there, no? Okay, here callus it is there. The callus generally so dissolved by calyx enzyme. Callus cells dissolved by calyx enzyme. Then automatically anther will be open. Then pollen grains will be releasing outside. Pollen grains will be releasing outer side. So that is the helps to the calyx enzyme. So next one it is also nourished to the pollen grains. Nourished to the pollen grains. Okay, nourished to the pollen grains. Okay, nourishment. It is also nourishment. It is also nourishment to the pollen grains. Okay, so it providing pollen grains. So to develop the pollen grains, how it will be developed? So it providing food to the nourishment to the pollen grains. Then pollen grains will be so developed. Okay, so this is called as a tapetum. This is called as a tapetum. So this is the so all layers. Okay, total how many layers are there? 4 layers. See here we are discussing what? So here we are discussing epidermis. Okay, epidermis. Next layer it is called as a so endothelium. Okay, this is endothelium. This is called as a callus. Callus. It is made up of callus. Okay, it is made up of callus. This is also callus. It is generally callus. Okay, callus. Okay, this is the callus. First we will discussing epidermis. So epidermis it is a outermost layer. Epidermis is a outermost layer. This is the outermost layer. Then thin walled cells. These are all are thin walled cells, not thick walled, thin walled, flat. How it is there? Flat shape, and it is also protective in nature. So next layer it is endothelium. So endothelium generally what it is there? Endothelium. So second layer. Endothelium is followed by, so generally epidermis is followed by endothelium. Next, radially arranged cells. How it is there? Radially it is arranged, these cells. And these cells with what it is there? Callose. So it is made up of, it contains callose, it is there. It is also helps to the dehiscence. The callose, it is also helps to the dehiscence of cells, means it is opening. And middle layer, next layer it is called as a middle layer. It is a thin walled cells and parenchyma cells. It is also helps to the storage of food material. It is also helps to the storage of food material. Next one, it is fourth layer. So this is the fourth layer. So generally it is a, so innermost layer, pyramid shaped cells. It contain a dense cytoplasma, more than 2 nuclei it is there. So then it is called as a multinucleated. The condition is called as a multinucleated. So due to the endomitosis, the mitosis where it is takes place inside, that's why, so it produces so number of nuclei it is producing. And formation of callus, it is also formation of callus. Nourishment to the pollen grains, okay, nourishes to the pollen grain, it is providing food to the pollen grains. Okay, so these are all are, so general characters. This is the, so general, these all, okay, what it is there? Epidermis, endothelium and middle layer and tapetum. So the wall layers of anther, okay. So next one it is here we will discussing so center position, okay. So this is the, where it is center, this is the center, okay. What type of tissue it is there? Homogeneous tissue it is there. So that is called as a homogeneous, all same type of cells, that is called as a sporogenous tissue. That tissue is called as a sporogenous tissue, okay. So this is called as a, so microsporangium, this is called as a microsporangium. So this is the microsporangium structure, okay. Microsporangia, this is one microsporangia, here one microsporangia it is there, here microsporangia, here microsporangia. So already yesterday we are discussing, this is the one lobe, this is the another lobe. So first lobe with 2 thecas are there, this is one theca, this is the one theca. So total how many? 4 thecas, that's why it is called as a tetrathecus condition. So inner side what layers are there now? Outer layer it is called as a epidermis, next layer it is called as a endothelium, next it is followed by a 2 to 3 layers are there, that is called as a middle layers, next it is followed by a tapetum it is there. So next so this region it is called as a, so microsporangia. The microsporangia center position cells are there, that is called as a sporogenous tissue. The sporogenous tissue only it is act as a microspore mother cell or pollen mother cell. Microspore mother cell or pollen mother cell. So this is all we are discussing, so microsporangium, okay. This is all we are discussing, microsporangium. So next we will discussing what is microsporogenesis. What is microsporogenesis? Microsporogenesis we will discussing here, okay. So next we will discussing microsporogenesis. Here we will discussing, next we will discussing microsporogenesis. So next what we will discussing? Microsporogenesis. So next topic is microsporogenesis. Next one it is microsporogenesis. Okay, next we will discussing microsporogenesis. What is microsporogenesis? Microsporogenesis is nothing but formation of generally it is the process of formation of, okay, it is the process of formation of microspore mother cell from the so microspore mother cell only. Okay, so here so it is the process formation of microspores, formation of microspores is nothing but pollen grains, pollen grains from the microspore mother cell, from microspore mother cell, microspore mother cell or pollen mother cell, pollen mother cell. It is also known as a sporogenous tissue or sporogenous cells. Okay, we will take one cell now, that's why sporogenous cell. Okay, here what we are discussing, so microsporogenesis. What is microsporogenesis? Formation of microspores. What is microspores? Pollen grains or male gametophytes form from the microspore mother cell or pollen mother cell or sporogenous cell. Okay, where it is there sporogenous cell? So this is the sporogenous tissue. Okay, this is the sporogenous tissue. If you take one cell, this is everything, it is a tissue. Okay, this is called as a sporogenous tissue. So if you take one tissue contains cells are there, no? Many cells are there, no? One of the cell, this is the one cell. That cell is nothing but what? MMC. Already we are discussing now microspore mother cell, or it is called as a PMC, pollen mother cell. Pollen mother cell. So MMC or pollen mother cell, it undergoes by the mitosis. Okay, by the mitosis. Okay, what it is producing? Okay, sorry, it is not mitosis. So one MMC undergo meiosis. Okay, so meiosis, this is the meiosis 1. First it is meiosis 1, it is so what it is? Reductional division. It produced a dyad. Okay, 2 pollen grains are producing. Okay, 2 pollen. Okay, that is called as a microspore tetrad. Oh, sorry, microspore dyad. Okay, it is microspore dyad. Next it is produced. So another meiosis. Meiosis is so generally, so meiosis 1 and meiosis 2. In second meiosis, how many are producing? Totally microspore tetrad. Okay, this is the total. How many are producing? 4 are producing. Then the pollen grains will be releasing. It is released into the, when anther is completely, it is matured, then it will be released. Okay, so where it is their pollen grain? So after microsporangia, generally after maturation, it is converted into the pollen sac. It is converted into the pollen sac. So now this is considered as a, so this is considered, generally it is a microsporangium. It is a fully matured. When it is fully matured, what happened? So then it is converted into the, so this is generally what it is? Microsporangia. This is nothing but microsporangia. When anther is mature, when anther is mature, converted into pollen sacs. Pollen sacs with what is there? Pollen sacs with pollen grains. The pollen grains, anther is completely, it is mature, then the pollen grains will be releasing. Okay, so releasing. Okay, releasing pollen grains releasing, okay, into the atmosphere. Okay, this is also we will see in. Okay, generally this is the microsporangia. Okay, microsporangia, when anther is matured, so it is converted into the pollen sac. The pollen sac with what it is there, pollen grains. The pollen grains released into the, where it is releasing, it is released into the, so atmosphere. Okay, it is released into the atmosphere, it is releasing. Okay, this is also you should remember. Okay, this is the generally before what it is there, young anther contain what it is there, you should write here young anther contain, young anther contain what it is there, microsporangia. So when it is anther is matured, it is converted into the pollen sac. The pollen sac with what it is there, pollen grains are there. The pollen sac with what it is there, pollen grains are there. You understood this one? Okay, see here. So generally, so this is the MMC. So MMC is nothing but formation of microspores or pollen grains. So generally where it is, from the microspore mother cell or pollen mother cell or sporogenous tissue. So this is also MMC, first meiosis, second meiosis, it produces pollen grains. It produces pollen grains. How many pollen grains are producing? So total 4 pollen grains are producing. It is released into the outer side. It is released into the outer side. Okay. So next, so this is the so next one it is microsporogenesis. Microsporogenesis is nothing but what? So formation of microspores from microspore mother cell. Okay, this is tissue. This is sporogenous tissue. One of the tissue contain many cells are there. No, one of the cell only it is act as a microspore mother cell or pollen mother cell. The pollen grain, first meiosis and second meiosis, total how many producing? So 4 pollen grains are producing. So this pollen grains, it is released into the outside. Okay, when it is fully it is matured, it is dissociated and released into the atmosphere. So this is also we are observing microsporogenesis. Microsporogenesis again we are discussing. So next again we are discussing. Next we will discussing so microsporogenesis. Okay, microsporogenesis already we are discussing that one only. Microsporogenesis, see here, this is the one of the microspore mother cell. This is nothing but MMC, or it is called as a PMC, or it is called as a sporogenous tissue. Okay, sporogenous cell, sporogenous cell. So this cell it is undergo. Okay, this cell it is undergo. So first meiosis, generally, so I will write here meiosis. Generally meiosis what happened? Generally next one it is undergo meiosis, it producing, here say meiosis. In meiosis, so what happened? Meiosis, we will discussing here meiosis. In meiosis, this is the one cell, first meiosis it producing 2 cells. Okay, 2 cells. Next, second meiosis, it is producing 4 cells are producing, total 4 cells. Okay, so this first meiosis is reductional division. The chromosome number will be reducing. So next division what it is? Okay, so next equational division, that is called as a equational division. Okay, one it is reductional division. Okay, it is reductional division. Another one it is equational division. Okay, equational division, you should remember this one. Okay, so generally it is a meiosis 1. This is meiosis 2, it is takes place. Here first we will discussing microspore mother cell or pollen mother cell. So first undergo, it is divided, first meiosis it producing how many cells? 2 cells. So this is called as a microspore dyad. Generally two only, no, dyad, microspore dyad, two cells are formed. Okay, so this is considered as a meiosis 1. Already we are discussing now that one only. Meiosis first it is reductional division, reducing the chromosome number. So next meiosis what happened? This is the meiosis 2. It is a equational division, equally distributing chromosomes. Okay, so now how many cells are producing? Totally 4 cells are producing. Total how many cells are producing? So 4. Okay, flow 4 are how it is there? Cluster of pollen grains. It is a cluster of pollen grains. Okay, it is a cluster of pollen grains. It is nothing but so microspore tetrad. This condition is called as a microspore tetrad. Okay, it is microspore tetrad. Okay, how many 4 pollen grains are there? So when so if it is fully matured no anther, then it is here callose. Before we are discussing callose. Okay, so generally it contain what it is there? Callose it is there. Callose is dissolved by, this is dissolved by, so generally it is dissolved by callase enzyme. Okay, callase enzyme it is dissolving, then automatically what will happen? This pollen grains will be so separating. Okay, it is dissociated. Okay, the pollen grains will be dissociated. Okay, so this is the formation of pollen grain. This is the formation of pollen grain. Pollen grains will be dissociated. Dissociated means, so now it is a cluster, then it is fully anthered, it will be mature, it will be dissociated. See one more time, microsporogenesis, formation of microspore from the microspore mother cell or pollen mother cell or sporogenous tissue. So this is the MMC, microspore mother cell. First meiosis is nothing but reductional division. Second meiosis is nothing but equational division. First dyad, 2 cells are producing. Next one is 4 cells, that is called as a microspore tetrad. Finally it is releasing into the pollen grains. Pollen grains will be, so all are clustered now, it will be dissociated, released into the atmosphere. Okay, when it is anther is fully matured, the what happened, this pollen grains will be callose is there. The callose will be break down by the callase enzyme, then pollen grains will be dissociated. Okay, so this is the microsporogenesis. Okay, so microsporogenesis. Next class we will discussing. So next what we are discussing? Pollen grain. Okay, it is nothing but what? Pollen grain. This is nothing but pollen grain formation. Okay, so now pollen grain. Tomorrow we will discussing what is pollen grain, how it is developed. Okay, development of male gametophyte or development of pollen grains. Tomorrow we will discussing. Okay, ma, bye.